Bioelectricity. Electricity in our body results from tissue positive and negative charges. Voltage gradient may act against concentration gradient. For example, sodium pump acts to pump sodium outward although intracellular sodium concentration is lower, and to pump potassium inward, although extracellular potassium concentration is lower. This process requires energy that is derived from adenosine triphosphate, ADP. This table shows that intracellular sodium concentration is lower than the extracellular, while extracellular potassium concentration is lower than the intracellular. Despite that, the sodium pump acts to pump sodium outward, and to pump potassium inward, against their concentration gradients. Sodium pump, sodium-potassium pump. The sodium-potassium pump, or simply the sodium pump, is an enzyme called sodium-potassium adenosine triphosphatase. Sodium pump has a particular significance for excitable cells, such as nervous cells, to respond to stimuli and transmit impulses. Sodium pump cycle. For each ADP molecule consumed, 3 sodium ions are pumped out and 2 potassium ions are taken in. The ADP-bound pump binds 3 intracellular sodium ions. ADP is hydrolyzed, and the pump is phosphorylated. A conformational change in the pump exposes sodium ions to the outside, which are released, because the phosphorylated form of the pump has a low sodium affinity. The pump binds 2 extracellular potassium ions, which causes dephosphorylation of the pump, returning it to its previous conformational state, transporting potassium into the cell, because the non-phosphorylated form of the pump has a low potassium affinity. ADP binds, and the process starts again. This is a schematic to show the 3 sodium, and the 2 potassium sites, in the sodium pump. What is the difference between the sodium channel and the sodium pump? Sodium channels transfer sodium passively down its concentration gradient, into the cell, inward current, and result in depolarization. This process does not need energy. Sodium pumps extrude sodium out of the cell, in exchange for potassium into the cell, against their concentration gradients. This process uses energy from ADP. Sodium pump functions. 1. Transport. Export of sodium from the cell provides the driving force for several membrane proteins, which import glucose, amino acids, and other nutrients into the cell. Movement of sodium from one side of a cell membrane to the other side creates an osmotic gradient that drives the absorption of water. Another important task of the sodium pump is to provide a sodium gradient that is used by certain carrier processes. In the gut, sodium is transported out of the cell on the blood side via the sodium pump, whereas the sodium glucose importer uses the created sodium gradient as a source of energy to import both sodium and glucose, which is much more efficient than simple diffusion. In the renal tubular system, similar processes occur. 2. Controlling cell volume. Inside the cell, several proteins and other organic compounds exist, and cannot escape from the cell. Most of these substances, being negatively charged, collect around them positive ions. These substances tend to cause osmosis of water into the cell, which can cause cell swelling and lysis. Sodium pump prevents cell swelling. Sodium ions, after pumping, tend to stay outside, creating an osmotic pressure to drive the water molecules out of the cells, since the membrane is less permeable to the larger hydrated sodium than the smaller hydrated potassium ions. When the cell begins to swell, this automatically activates the sodium pump, which moves more ions to the exterior. 3. Signal transduction. In addition to the classical ion transport, sodium pump can also relay extracellular signaling into the cell through activation of mitogen-activated protein kinase signal cascades, activation of mitochondrial reactive oxygen species production, activation of phospholipase C, activation of inositol triphosphate receptor. Thank you.